Hello, welcome back guys, it's Ryan Ling. Today, lecture 11, molecular genetics, session 2. Okay, question 1. We have a virus here. I know this is a bacteriophage from the look of it, right? So it has the head part, which is made of the capsid, right? And this is a neck, and here we have legs. Okay, number 1, which structure attaches this organism to the cell surface? Okay, the cell surface, they have these elegant legs, right? So this is the part that land on the surface of the bacteria and, detect a specific amino acid sequence or the cell membrane sequence, right? Okay, so it's, what's the number? Number 3, right? C. Number 2, structure 1 can be made up of, what is structure 1? This is inheritable materials, right? So for the virus, it can be either DNA or RNA, right? So both nucleic acid works, but here we don't have the RNA, so the only option for us is DNA. So A. Okay, number 3, structure 2 can be made up of, 2 is capsid, right? And we know that capsid only consists of protein. So 3, which gives us C, right? Okay, number 4, given the following single strand of DNA sequence from GAA-TTC, start from the 5' to the 3' ending, right? What is the sequence of the opposite strand? Okay, so 5', GAA-TTG, just write it down, okay? You always have to have anti-parallel double-stranded DNA, right? So for this one, it's got to be CTTAAC, right? You cannot use a uracil because it is a DNA sequence. Okay, but here, in all the options, we have it in from 5' to 3' direction. So we got to flip it. So CAA-TTC. Where is it? CAA-TTC. So A. Number 5, which of the following will not be present in a molecular messenger RNA? Okay, mRNA, it is RNA, so you cannot have thymine because you can only find this nucleotide in the DNA, right? Okay, number 6, which of the following occurs in the cell nucleus? Okay, number 1, RNA synthesis, which is same as the transcription, right? And the protein synthesis takes place in the ribosome, which resides in either cytoplasm, or RLVR. So it's not. Okay, number 3, DNA synthesis, of course, takes place in the nucleus. So 1 and 3 gives us D. Okay, let's go to number 7, purine that binds with 3 hydrogen bonds. Okay, so for the purine, you have adenine and guanine. And for the pyrimidine, you have cytosine, thymine, and for the RNA, uracil, right? So in this case, purine that binds with the 3 hydrogen bond. If you remember, adenine always pairs up with thymine, right? And you can see 2 hydrogen bonds between A and T. But between the C and G, you can see 3 hydrogen bonds, right? So that means the bonds or the relationship between the cytosine and the guanine is a little more stable than the A and T, right? More stronger bonds. So since it's talking about the 3 hydrogen bonds, it's got to be between the C and G, and purine, in this case, is guanine. So B is our answer for the number 7. Okay. And next one, found only in RNA. It's talking about uracil, right? Number 9, pyrimidine that binds with the 3 hydrogen bonds. So it's talking about the pyrimidine, so it's got to be cytosine. Number 10, purine found in both DNA and RNA, they bind with the 2 hydrogen bonds. What is it talking about? Adenine, right? So it's A. Okay, number 11, which of the following is true regarding DNA replication? Okay, number 1, DNA replication is semi-conservative. Yes. Number 2, DNA replication occurs during prophase. No. Before prophase, right? Takes place during S phase of interphase. So number 3, DNA synthesis occurs in a 3 to 5 prime direction. Definitely not. Number 4, purines hydrogen bond to pyrimidines. Okay, purines to pyrimidine is correct, right? Because adenine, which is purine, always pairs up with the thymine, which is pyrimidine, and guanine, purine, pairs up with cytosine, pyrimidine, right? Purine, pyrimidine, purine, pyrimidine. So 1 and 4 are the correct statements. 1 and 4. So E. Okay, number 12. Wait a minute. All thymines of a double helix is radioactively labeled. After some time, the cell is induced to undergo 2 rounds of replication. Okay, 2 rounds of replication, and it is already radioactively labeled or tagged, right? So, okay, so I'm gonna draw this double helix in yellow if it's radioactively labeled, and after one round of replication, it becomes like this, and after second round, becomes like this. 1, 2, 3, 4. Okay, scatter to 12. Some of the radioactivity will appear in half of the resulting double helices, right? So B is correct. And for 13, the ratio of the original radioactivity to the resulting radioactivity will be, okay, the original one concentration, if we say is 1, then after this, 1, 2, 3, 4, 5, 6, 7, 8. Out of the 8 single strands, only 2 of these are radioactively labeled. So it's got to be only quarter of the original radioactivity, right? So it's got to be 4 to 1. So C. Okay, 14. Which statement regarding protein synthesis is false? Okay, we got to find the false one. A, tRNA molecules help incorporate the correct amino acid into proteins. Yes, it is correct, right? And B, proteins are formed on the ribosomes, correct? C, mRNA is not necessary for proper protein synthesis. Definitely not, because mRNA is the one that acts as a template, and without it, we cannot do anything. We cannot make protein. D, rRNA is needed for proper binding of the mRNA message. This is correct, right? Because for the ribosome, it binds everything. So first it binds mRNA, and then it binds tRNA, right? And at the same time, it binds polypeptides, right? So without rRNA, without ribosomes, you cannot, dissociate between the mRNA and tRNA.